In the summer of 1947, a young shepherd searching for a lost goat in the hills above the Dead Sea threw a stone into a dark opening in the rock. Instead of a hollow echo, he heard something shatter. Curiosity pulled him inside, and what he found there would eventually reshape the study of the Bible itself. Tall clay jars, sealed for 2,000 years, holding brittle scrolls of animal skin parchment wrapped in strips of linen. That single accident opened the door to the Dead Sea Scrolls, the oldest surviving copies of Hebrew scripture ever found, and one of the most contested archaeological discoveries of the modern age. For decades, the scrolls were treated as a kind of sacred puzzle box. They had not survived the centuries in one piece. Time, moisture, and decay had reduced them to tens of thousands of fragments, many no larger than a fingernail, scattered across 11 different caves. There were no covers, no page numbers, no way to know at a glance which sliver of leather belonged with which. Scholars had to reconstruct entire manuscripts using nothing more than handwriting style, ink color, and the texture of the parchment itself, a method that was more art than science, and one that was bound to make mistakes. Those mistakes mattered enormously, because the stakes were not academic trivia. The scrolls include the earliest known copies of books from the Hebrew Bible, predating the next oldest surviving manuscripts by nearly 1,000 years. Every choice about which fragment belonged to which scroll shaped how the modern world understood the origins of Judaism and Christianity alike. And for much of the 20th century, those choices were made by a strikingly small circle of scholars who controlled access to the material, publishing findings on their own schedule, and, for long stretches, keeping much of the collection hidden from outside review. It would take more than 3 decades before the full body of scrolls was finally made available to researchers worldwide, and by then, a great deal of scholarly consensus had already hardened around theories that nobody could fully verify. The dominant theory, repeated in textbooks and museum placards for generations, held that the scrolls were the library of a reclusive Jewish sect called the Essenes, who supposedly lived an ascetic, communal life at the nearby settlement of Qumran and hid their books in the surrounding caves when Roman legions threatened the region. It was a tidy story. It was also built on remarkably thin evidence. The word Essenes never once appears in the scrolls themselves. Everything scholars thought they knew about this group came from 2 outside writers, the historian Josephus and the Roman naturalist Pliny the Elder, neither of whom claimed to have lived among the group they were describing. Some modern historians have gone so far as to argue that the Essenes, as a distinct, definable sect, may never have existed in the form later assumed, that the label was a convenient shorthand invented by outsiders and then adopted wholesale by 20th century scholarship because no better explanation was on offer. For 70 years, there was no way to test any of this directly. Handwriting can be imitated. Parchment can look similar across very different sources. The field had reached the edge of what traditional methods could tell us, and it stayed there, until a chance conversation on a university shuttle bus changed everything. In 2012, 2 professors at Tel Aviv University found themselves seated next to one another during a new faculty orientation. One was a molecular biologist who spent his career studying genetic material. The other was a specialist in ancient biblical texts who had spent years wrestling with the unsolved puzzle of the scroll fragments. As the biblical scholar described the scale of the problem, tens of thousands of disconnected pieces, no reliable way to match them, and a real risk that mismatched fragments had already been stitched together into manuscripts that never actually existed. The biologist asked a simple question: what is the parchment made of? The answer was animal skin. And animal skin, unlike ink or handwriting, carries something no forger or careless conservator can fake: DNA. Every living creature leaves behind a unique genetic signature embedded in its hide. In theory, if 2 fragments came from the same animal, their DNA would match. If they came from different animals, different herds, different flocks, different regions entirely, the genetic evidence would say so with a certainty no paleographer could offer. It was, on paper, the most promising idea to enter Dead Sea Scrolls research in half a century. It also came from someone who had never studied an ancient religious text in his life. Turning the idea into practice was another matter. The scrolls are treated as irreplaceable religious and historical artifacts, and no one was willing to cut into them or scrape material from their surface to extract usable samples. Every standard method of ancient DNA extraction required exactly the kind of physical sampling that was off the table. The breakthrough came from an unglamorous source: dust. Over decades of storage and handling, microscopic particles of parchment had naturally flaked away from the fragments and settled at the bottom of their protective cases. That dust, invisible without magnification, still carried traces of the original animal skin material, without requiring anyone to touch the scrolls themselves. Collecting and processing that dust demanded extraordinary care. Researchers worked in sealed, positive-pressure laboratories of the kind normally reserved for the most sensitive biological research on Earth, sterilizing every surface and instrument with ultraviolet light between uses. Even then, the dust was contaminated, not just by the ancient animal it came from, but by every human hand that had touched the scrolls over 70 years of study, conservation, black market trading, and forgery. Scientists had to computationally filter out layers of modern human DNA just to isolate the faint ancient signal buried beneath it. And the ancient material itself was in poor condition to begin with. The parchment-making process had already fractured much of the original DNA. The iron-based ink used to write on the surface had corroded genetic material on contact, and 2,000 years of heat, cold, and mineral exposure inside desert caves had destroyed still more. It took roughly 7 years of laboratory work, hundreds of samples, and painstaking genetic sequencing before the team had results they trusted enough to publish. What they found rewrote assumptions that had gone unchallenged for generations. The first surprise was almost mundane on its own. Nearly all of the tested fragments turned out to be sheepskin rather than goatskin, meaning that decades of matching fragments by feel and appearance had been done using the wrong reference material from the start. It was an unglamorous correction, but it exposed just how much of the old methodology had rested on assumption rather than evidence. The far more consequential discovery involved 2 fragments that came back not as sheep or goat, but as cowhide. That single detail carries enormous weight, because cattle could never have been raised in the arid landscape around Qumran. Cows require large volumes of fresh water and open grazing land every day, resources the Judean desert simply does not have. If those 2 fragments were made from cowhide, they were not produced anywhere near the caves where they were found. They had to have originated somewhere with real pasture and a reliable water supply, most plausibly a fertile region closer to Jerusalem, roughly 20 miles away. That finding alone was enough to undercut the old sectarian theory. If some scrolls were manufactured near Jerusalem and later transported into the desert, then the caves were never simply the private archive of one isolated desert community. Someone carried those documents a considerable distance, under conditions that strongly suggest urgency. The most obvious historical trigger is the Roman siege and eventual destruction of Jerusalem in 70 CE, when the city burned and its population scattered. It is a sobering thought. The DNA cannot tell us who carried those scrolls into the desert, whether they were fleeing alone or with family, or whether they lived to see another year, only that someone, under pressure, judged those particular texts worth saving. The genetic analysis also exposed something scholars had noticed before, but had never been able to prove conclusively. One of the cowhide fragments had, in earlier scholarship, been physically joined to a separate sheepskin fragment and treated as part of a single continuous manuscript. The DNA evidence made clear that these 2 pieces came from different animals raised in different regions. They could not plausibly belong to the same original scroll. A document that had been published, cited, and studied as one coherent ancient text turned out to be a modern reconstruction error. 2 unrelated fragments fused together by well-intentioned but mistaken scholarship. Both of those mismatched cowhide fragments happen to come from the book of Jeremiah, and that detail opened the most striking finding of the entire study. Biblical scholars have long known that 2 distinct versions of Jeremiah exist, a longer version that matches the Hebrew text used in the modern Bible, and a noticeably shorter version, missing entire passages and arranged in a different chapter order, that closely resembles the version preserved in the ancient Greek translation known as the Septuagint, which shaped the Old Testament used by early Christian communities. For years, Scholars debated why these versions differed: scribal error, later editing, or something more fundamental, without a way to settle the question. The genetic evidence tipped the balance. Because the two Jeremiah fragments came from animals raised in different regions, it strongly suggests these were not simply two copies of one evolving text, but two separate textual traditions, developed and maintained independently by different communities, quite possibly unaware that the other version even existed. Both communities, presumably, regarded their version as the authentic word of Scripture. At the time these scrolls were produced, in other words, the Bible was not yet a single, fixed document. It was still taking shape, existing in parallel forms across different communities, until later editorial decisions, made by people whose reasoning is now lost to history, determined which version would be copied forward and which would eventually be set aside. The version that did not survive into the mainstream tradition is partially preserved only because fragments of it happened to end up in a desert cave. Taken together with the broader genetic patterns across the collection, clusters of closely related animals suggesting local production in some cases, alongside fragments with no genetic relationship to anything else nearby, the DNA data support an idea that some scholars had proposed but never been able to confirm, that the caves were not the private library of a single isolated group, but something closer to a communal repository, a geniza, where multiple communities across the region deposited sacred texts too worn or damaged to use, but too holy under Jewish law to destroy. Layered on top of that ordinary religious practice were, it seems, the emergency deposits of people fleeing violence in 70 CE, adding their own most treasured scrolls to a collection that had likely been accumulating for generations. It's worth being honest about the limits of what this research actually proves. The genetic study examined only a small fraction of the roughly 25,000 fragments recovered from the caves, a tiny sample set against the full scale of the collection. From that modest sample, researchers were able to challenge a theory that had shaped the field for 70 years, but enormous amounts of material remain untested, and some fragments are now too degraded for genetic analysis to ever recover anything from them. Some texts within the wider collection were also written in deliberately obscure scripts that took decades to decode, and scholars still debate why their authors felt the need to conceal their contents at all. DNA can identify an animal's species and rough geographic origins. It cannot tell us the name of the person who carried a scroll out of a burning city, whether they survived the journey, or what they believed would happen to the words they left behind in the dark. What the research does make unmistakably clear is that the story scholars told for most of the 20th century, a single reclusive sect, quietly copying scripture in isolation until Rome forced them to bury their library, was too simple to be true. The real picture that is emerging is messier and, in some ways, more human. Multiple communities across ancient Judea, each producing and safeguarding their own sacred texts, converging on the same caves through both ordinary religious custom and the desperate improvisation of people running from a war they could not stop. What survived in those jars was never a complete record of ancient Jewish religious life. It was a fragment of a much larger, more varied world, most of which was lost long before anyone thought to sequence its DNA. The shepherd who threw that stone in 1947 had no way of knowing what he had stumbled into. He had found the last physical trace of communities that vanished when Jerusalem fell, voices that had been silent for 2,000 years, waiting in the dark for someone with the tools to finally hear them. Modern genetics gave those voices back, at least in part. But as the scientists themselves acknowledge, what has been recovered is only a fraction of what once existed. The scrolls that made it into those caves survived by chance. Far more, in all likelihood, did not survive at all, and what was lost with them may be greater than anything the caves managed to save.